Hi, we will be discussing some of the important aspects of the different organ systems in the body, how they work, how they coordinate. It's fascinating how our body works, so it's really a magic of life. Basic genetics. So, as we know, the information that codes the proteins for our body, the information that decides how we function as an individual, is carried on our genes. And the genes have genetic material in our DNA, which is a double stranded deoxyribonucleic acid. This is carried on chromosomes. There are millions of genes, and they are carried on the different chromosomes. 46 chromosomes in our body in each cell, 23 pairs, which include 22 autosomes and one pair of 6 chromosomes. So this arrangement is called diploid because we have pairs of chromosomes. The sex chromosomes includes the X and the Y chromosome. The male has X and Y, and the female has two X chromosomes. The normal body cells multiply by mitosis, which results in duplication of the genetic material into two equal halves. The diploid pattern is preserved. Meiosis is specific for division of the gamete cells, which is the sperm and the ovum. This results in splitting of the genetic material into half. So each sperm or each ovum has only one half of the genetic material needed for a cell. This arrangement is called haploid, and this haploid nature of the gametes is responsible for gene mixture between the two parents. So the genes come together, one half from each parent, and there is mixing of genetic material during the process. This is an illustration of the DNA structure, how it then coils around, and basically the coils become chromatid bodies, which are all contained in the chromosome. The junction of the chromosomes limbs is called centromere, and we have the short arm or the petite arm, P for petite, and we have the long arm, which is also called the Q arm. So the long parts of the segments are called the telomeres after the centromere. And this is an illustration of how it looks like on the karyotyping, which is basically a spread of the genetic information, the chromosomes. So we have chromosomes numbered 1, 2, 3. They have a banding process, and depending on how the bands are distributed, the length of the arms, the position of the centromere, the chromosomes are categorized. So we have 22 pairs of autosomes and the sex chromosome. In this illustration, it's 2 X chromosomes. So the genetic material, the genes have exons, and the chromatid bodies have exons and introns. The exons are the coding part, and the introns are supporting parts. They do carry some information which are called the epigenetic factors, which can influence the gene expression. The exome is a part of the genome which codes, which is a component of all the exons, and the whole genetic information, including the exon and intron, is called the genome. So these words become important when we look at the recent tests available for assessing genetic material, which is the whole genome sequencing and so on. When we call epigenetics, it's the different factors that can influence the way the genetic information is expressed. For example, two individuals might have the exact same genes, but they manifest differently in different functions because of the different epigenetics. So the epigenetics can happen from external factors, or it may be present in your environment. The epigenetic factors that happen during pregnancy and during the delivery process in the first week after life is very important, and these impact the lifelong manifestations. The gut microbiome also has a very important role in the way the epigenetics is reflected. So what bacteria we have in our gut may influence how we respond. This also explains family traits where the parents and the baby may carry the same gut microbiota, or the predominant pattern may be similar and they express similar patterns. We should be familiar with the modes of inheritance, and you may also call that the Mendelian inheritance model, because Gregor Mendel gave the structure for this. So we have autosomal recessive conditions where we need two abnormal genes to come together to manifest, to be deficient to manifest, and usually these are the metabolic or enzyme related conditions. We can have the autosomal dominant condition where even one abnormal gene can manifest, and this is usually variable penetrance, which means the presentation can be different in different individuals, or different generations may be affected differently. Most of the autosomal dominant conditions are structural conditions related to cartilage, bone development, and so on. And we have the X-linked disorders, which are specific in that the severe condition is only in the males. The females may have a milder form or a carrier form. So both in autosomal recessive and in X-linked conditions, we have a carrier form which doesn't manifest the full disease, but when two carriers come together, you have the disease in a good proportion. We also have additional chromosomes which manifest as trisomies. So we saw in the karyotype how we have 23 pairs of autosomes. If you have one extra chromosome, then it's a trisomy, and this extra chromosome can be chromosome 21 in Down syndrome, chromosome 18 in Edwards syndrome, and chromosome 13 in Patau. We also have conditions like deletions, duplications, and translocation of genes. There is a condition called genomic imprinting, where only one half the paternal version of the gene is expressed, or the maternal and you may have condition like Prader-Willi syndrome resulting from that. So for basic understanding, you can be aware that mutations happen in the genetic material, and if it happens in the gametes, then it can be transmitted. If it happens in the body cells, they may not manifest in that, but you may lose their ability to control the regulation of multiplication. So tumors may happen as a result of mutation in the somatic or body cells. And trisomies are very common. So you have Down syndrome, which has typical features. So if you see one child with Down syndrome, you would be able to pick up this pattern. Recognizing the pattern is called gestalt recognition, and we don't need to do any tests. Once you've seen a few children, you know that this is Down's. Some of them are borderline, and we may need to review that as well by genetic testing. So you have single palmar crease. This can be normal as well in 3%, so not everyone with a single palmar crease has Down syndrome. White spots on the colored part of the eye, which is the iris. We have small ears, small mouth, and flattened nose, and the back of the head is flat as well. And the fingers are short and stubby, and the muscle tone is reduced. There may be a wide sandal gap, as we call wide gap between the toes. These children are at risk of different conditions like leukemias. Heart disease is very common. Gastrointestinal abnormalities are common as well. This is an example of one of the commonest trisomies. The incidence is about 1 in 700 to 1 in 800. And very typical feature here is most of these happen due to non-meiotic disjunction, which means the two halves fail to separate during the meiosis, which results in the ovum formation. The risk of this happening increases as the mother gets older, and so as the mother's age exceeds 35 years and especially beyond 40 years, the chances of Down syndrome increase rapidly, and it may fall to as low as 1 in 50. Normally I said it's 1 in 800, but it starts dropping from 35 years onwards, and by age of 45 it reaches as low as 1 in 50 of risk of Down syndrome. So the higher the age, we offer antenatal screening to look for certain markers, and you can also do genetic testing on samples elicited from either chorionic villus sampling or amniotic fluid sampling, which can result in confirmation of the karyotype. So in the next presentation we'll look at basic embryology, and I hope this short presentation is useful. Thank you.